Hello everyone, and thank you for joining us for our LIPID MAPS webinar this week. I'm Ruth Andrew, your host, and I'm from the University of Edinburgh. And with no more ado, I'd like to introduce our guest speaker, Dr. Mark Nixon, who I'm delighted is joining us today. Mark and I have the pleasure of working together for some time. He did his PhD studying in my group, where he investigated the role of glucocorticoid metabolism in inflammation. And this has laid the foundation for a research career focusing on the intersection of endocrinology, metabolism, and cardiovascular health. Mark has spent some time in the University of Texas Health Sciences Center in Houston working with Professor Rebecca Verdot, and there he discovered a novel role for the AMPK-related kinase SIK1 in regulating glucose homeostasis in obesity. He returned to Edinburgh in 2014, working with Professor Brian Walker's group, where again he uncovered a new function, this time for a transmembrane exporter, ABCC1, in controlling glucocorticoid access to adipose tissue. And all this laid the foundations for a BHF Basic Science Intermediate Research Fellowship, which Mark was awarded in 2018. And there the focus was on how cleavage of corticosteroid binding globulin influences adipose glucocorticoid delivery, again in the obese setting. So Mark is now a lecturer, University of Edinburgh. His group is involved with investigating many aspects of tissue-specific regulation of glucocorticoid action. And here they're trying to help redefine the field of how glucocorticoids in the circulation are not the only player. You have to look at the tissue, and also identify new opportunities for therapeutic interventions. He studies cells, animal models, and also human disease. So with no more ado, I'm delighted to hand over to Mark to deliver his lecture from Circulation to Cell, Steroid Trafficking in Cardiometabolic Health and Disease. So Mark, over to you. Thank you.
Well, thank you for that really kind introduction, and thank you also to LIPID MAPS really for the opportunity to talk with you all today. Thank you everyone for coming along, and really it's a pleasure to be able to share with you some of our continuously evolving research into the role that these stress hormones play in obesity and indeed cardiometabolic health and disease in general. So I thought let's really just jump right into it and start with obesity itself. And ask, is it really worth stressing over? And I suppose, unfortunately, it's a bit of a spoiler to say, yes, it is. Unfortunately, despite all our advances, obesity is still on the rise. And putting that into perspective, I suppose, is a study in The Lancet just last year that tracked prevalence of obesity over the past 30 years. And the results are really rather striking in adult men and women, and most worryingly in children and adolescents. Rates have more than doubled over the last 3 decades. And this raises major concerns, obviously, for long-term health trajectories across the lifespan. Because it's not just about the weight, it's what obesity leads to. It significantly increases the risk of developing a range of serious and often life-limiting conditions, including type 2 diabetes, hypertension, osteoarthritis, coronary heart disease, and at least 13 different types of cancer. And these aren't isolated problems. They often cluster together, placing a massive burden not only on individuals but also on healthcare systems as a whole. So in the UK alone, for example, the treatment of obesity and its related conditions was projected to cost the NHS £6.5 billion in 2022. And looking ahead, the World Obesity Federation projects that by 2035, the global cost of obesity will rise to $4.3 trillion annually, so a figure that's equivalent to around 3% of the global GDP. What does this tell us? Well, really it says that despite decades of research and countless public health campaigns, there still remains this huge unmet need. We urgently need to better understand the biological mechanisms that link obesity to these chronic conditions. And just as importantly, we need to develop more effective and targeted interventions that don't just manage the weight, but actually address the underlying drivers of the associated metabolic dysfunction. And that's really the focus of my research, if you like, to try and go beyond associations and unravel the molecular and tissue level pathways that connect obesity to cardiometabolic risk, and ultimately to find new ways to intervene. In particular, I want to give you a brief insight into our work on glucocorticoids and their role as a link between obesity and cardiovascular disease. This is a primary glucocorticoid here in humans called cortisol, and of course one of many steroid hormones that we produce in our bodies that form a subclass of lipids. In humans, cortisol is produced by the adrenal glands in response to stress, so under the command of the hypothalamic pituitary adrenal or HPA axis. And you can think of that as part of our internal crisis response system, if you like. So here cortisol helps us adapt and overcome some acute challenges. And that includes waking up in the morning, fasting, or fighting infection or dealing with inflammation. And its job here really is to restore balance, to bring the system back to normal. And once a crisis is over, cortisol levels fall. And what's really important to emphasize here is that in the short term, these acute responses of the body in activating the HPA axis and releasing cortisol are essential adaptive responses to survival. The issue, or the trouble if you like, is that if this high cortisol response is prolonged or maintained over the long term in a way that's no longer appropriate to the circumstances, what happens here is that all these adaptive responses become maladaptive. And we know this because our clinical colleagues see patients who have, for example, tumors that cause cortisol levels to be high all the time. This is called Cushing's. And these patients develop a whole host of clinical problems, including central or visceral obesity associated with diabetes, hypertension, abnormal cholesterol, or dyslipidemia. And hopefully, of course, you can see where I'm going with this, because these are all risk factors for cardiovascular disease. Of course, led to the hypothesis that within the general population, an activated HPA axis and thus increased cortisol levels may predispose some of us to developing these cardiometabolic risk factors and developing end-stage cardiovascular disease. And over the years, there has been a steady stream of observational studies linking cortisol and cardiovascular disease, with many showing that increased glucocorticoids, mainly iatrogenic in source, are associated with increased cardiovascular disease risk. However, it's unclear whether these observational studies' associations reflect true causal effects. So the gold standard method of determining causality is, of course, through randomized controlled trials. However, they are laden with issues including cost, time, ethical considerations. So finding alternative approaches is beneficial. And one of these is Mendelian randomization, or MR. And it's a statistical method analogous to a randomized controlled trial, except that in this case, genotypes are used to randomize participants into different exposures of interest. And in this way, Mendelian randomization enables causal relationships to be evaluated by examining the relationship between genetic susceptibility to the exposure or risk factor in question and the disease outcome of interest. So what I'm doing now is about to bundle probably a decade's worth of work into about a quarter of a slide, but I will revisit this, so don't worry. But really the take-home message is that in Edinburgh, we established the CORNET Consortium, which now includes data on morning plasma cortisol levels acquired across 17 studies of European ancestry with over 25,000 subjects. And the most recent publication from this Mendelian randomization identified for the first time that cortisol causally increases the risk of heart disease. And what I want you to do here is just pay attention, just remember, if you like, the genetic variant, which is SERPINA6. I'm going to come back to that a little bit later in my talk, but for now I want to focus on how we might intervene to limit glucocorticoid action. So for this, I thought I'd start by giving you an overview of how we currently treat HPA axis disorders. So I've already mentioned Cushing syndrome. Usually this results from a pituitary tumor secreting ACTH, which in turn drives excess glucocorticoid production, and therefore induces adverse, mainly peripheral consequences. And the treatment goal here is to restore normal glucocorticoid secretion. And the primary intervention is usually surgical removal of the ACTH secreting tumors. That leads to remission of these adverse effects. In congenital adrenal hyperplasia, there's a blockage in the assembly line that makes cortisol. Therefore, the resulting low circulating glucocorticoids means reduced negative feedback, ultimately driving constant ACTH stimulation of the adrenal gland. And what that means is that the precursors that make glucocorticoids are shunted into other pathways, mainly the one resulting in testosterone production. And this increased or excess androgens leads to separate adverse peripheral consequences. Now, the current treatment here involves administration of glucocorticoids to suppress ACTH and therefore also suppress androgens. However, there are disappointing long-term outcomes for CAH patients, which reflect either inadequate suppression of this androgen excess or excess exposure to these exogenous glucocorticoids. So really, in the interest of time, what research has led us to is that intervening at this kind of central level to limit glucocorticoid action is not really a viable option because, as I mentioned, of course, the stress response is crucial. But by manipulating glucocorticoids at a tissue level, we may be able to have some benefit. And in particular, we believe that adipose tissue holds the key to this aberrant glucocorticoid cardiometabolic action. And indeed, there have been multiple studies over the past 20 years that have demonstrated that increased glucocorticoid action, specifically in adipose tissue, can drive obesity and its associated comorbidities. So in my lab, we've been trying to unpick tissue-specific regulation of these glucocorticoid stress hormones. So in doing that, on one side, we've been looking at this in the setting of obesity, as I've already mentioned, a major cardiovascular disease risk factor, and that's associated with glucocorticoid dysregulation at a tissue level, particularly in adipose tissue. But we've also been interrogating this in the setting of exogenous glucocorticoid replacement therapy. Essential, as I've mentioned, for many patients, but that comes at a cost, and that's raising cardiovascular disease risk sixfold. So for today, I'm going to mostly focus on our work over the past couple of years on glucocorticoid trafficking in obese adipose tissue and how this contributes to insulin resistance. I want to highlight some of the people in my lab who've worked on this, but in particular Luke Boyle, who led the human study investigating this. But of course, we can't really determine where we're going without first knowing where we've been and how we got here. So I want to briefly spend a minute or two to go over what's known about some of the mechanisms that control intracellular glucocorticoid action within adipose tissue and within adipocytes. So if we consider this rather simplified cartoon of glucocorticoid signaling within adipocytes, we know, for example, that glucocorticoids diffuse into cells, binding their cognate receptor. For the purpose of this talk, I'm going to refer to that as the glucocorticoid receptor, but of course there's now evidence that activation of the mineralocorticoid receptor may be important in some of the glucocorticoid-mediated actions in specific adipose depots. Following binding, this ligand receptor complex translocates to the nucleus and induces transcription. And really our focus, and indeed the focus of many other research institutes, has been on pathways that control what's referred to as the intracellular glucocorticoid pool. Now, many of you will be familiar with pre-receptor metabolizing pathways that influence glucocorticoid action. In particular, I'm sure most of you will be aware of the 11-beta-HSD enzymes, which interconvert active glucocorticoids with inactive glucocorticoids. And in Edinburgh, this has been work that's been led by Brian Walker. Ruth Andrew here in Edinburgh has also led research into the metabolism of glucocorticoids by 5-alpha reductases, in particular 5-alpha reductase type 1 in adipose tissue. And more recently, Ruth Morgan has identified 20-beta dihydroglucocorticoid metabolites in adipose tissue, formed as a consequence of the action of the enzyme carbonyl reductase 1, or CBR1. But stepping away from pre-receptor metabolism, we now know that while glucocorticoids passively diffuse into cells, there are a set of transmembrane transporters, known as ABC transporters, that are capable of selectively exporting glucocorticoids across cell membranes. We now know, for example, that ABCB1 selectively exports cortisol from cells, so that's the primary human glucocorticoid, while ABCC1 selectively exports corticosterone, the primary rodent glucocorticoid. although it is also present in humans. And we identified that in adipose tissue, which is high in ABCC1 expression but low in ABCB1 expression, that preferential export of corticosterol occurs. And this initial work has been taken on by a clinical fellow, Katrina Kyle, and a postdoc, Elisa Villalobos, in the context of whether corticosteroid may actually be a metabolically safer glucocorticoid replacement therapy than cortisol, given its export from adipose tissue. And this is something we're now exploring further, using preclinical models as well to kind of better understand the role that ABCC1 may play in the differential effects of cortisol and corticosterol. And that's work being taken on by Marie Paul. So that's a brief overview of some of the key pathways involved in regulating the intracellular glucocorticoid pool. But of course, they all take place within the cell, if you like. And my interest has really evolved into taking a bit of a step back and looking at what happens outside of the cell, and in particular focusing on how glucocorticoids get delivered to adipose tissue. Now, as I mentioned, glucocorticoids are released from the adrenal glands into the circulation. But what happens next, how they reach and act upon specific tissues, remains something of a black box. We don't really fully understand how glucocorticoids are targeted or delivered to certain organs or tissues. So we started working on a fairly straightforward hypothesis that certain tissues like adipose may send out some sort of signal, a kind of molecular distress call, if you like, that says we need cortisol, and somehow the body is able to respond. So to begin answering this, it's important not to consider the intracellular glucocorticoid pool, but to consider the circulating glucocorticoid pool, if you like. And for those of you who were paying close attention earlier, you'll recall that from our early GWAMA analysis, I told you about the major genetic variant that controls circulating cortisol, and that was SERPINA6. And of course, that encodes a protein known as CBG. And why is that important to know? Well, CBG binds around 80% of circulating glucocorticoids. And that's critical because according to the free hormone hypothesis, only unbound or free steroids like glucocorticoids can actually leave the bloodstream, enter tissues, and exert biological effects. So hopefully already you can start to see how CBG functions as a kind of gatekeeper, tightly controlling how much active glucocorticoid reaches the tissues. But of course that still leaves us with a fundamental question. What's the mechanism or signal, if you like, that allows certain tissues like adipose to call in this stress hormone? To introduce a new cell, the neutrophil. Now most of us here will know neutrophils well. We'll know that when they're activated, they release a powerful arsenal of antimicrobial molecules from their azurophilic granules. And one of these is a protease known as neutrophil elastase, or NE. You might ask why I'm talking about neutrophils in the context of obese adipose tissue. Well, again, many of you will know that obese adipose tissue is known as a pro-inflammatory environment. And several seminal studies over a decade ago now helped uncover what was at the time a surprising role for neutrophils in that setting. And what was seen was early and rapid infiltration of neutrophils after just 72 hours into adipose tissue upon high-fat diet feeding, along with clear increase in local neutrophil elastase activity. And around the same time, another group showed that neutrophil elastase levels rise during obesity, and they actually generated neutrophil elastase knockout mice as well, demonstrating that these mice exhibited improved glucose tolerance. And this suggests that this protease may be more than just a bystander. So at this stage of our investigation, we were aware that glucocorticoid excess in adipose tissue promotes metabolic dysfunction, and we were also aware that increased neutrophil elastase activity in adipose also drives metabolic dysfunction. And so the question became, is there a link? Is there a proverbial red string, if you like, on the evidence board that connects these two seemingly separate suspects. And of course, it wouldn't be much of a story if there wasn't. And their accomplice, if you like, is none other than CBG, so the hormone's chaperone in the bloodstream. And that's because CBG is a proteolytic target of neutrophil elastase, so NE cleaves a specific site on the surface of CBG, and what that does is cause a conformational shift, often described as a move from a stressed to a relaxed state. And that cleavage reduces CBG's binding affinity for cortisol nearly tenfold. And the consequence of that is the release of the glucocorticoid payload at the site of cleavage. But of course, to date, the idea that this neutrophil elastase CBG axis regulates glucocorticoid exposure at sites of neutrophil activation hasn't been tested in vivo, so it's only been shown in vitro. So we took this on, and worked towards a hypothesis that in obese adipose tissue, neutrophil elastase mediated cleavage of CBG results in increased local glucocorticoid exposure, an effect that drives adipose dysregulation and insulin resistance. And today I'm going to walk you through some of the key results that we've uncovered. And we started by showing in serum isolated from mice that treatment with neutrophil elastase reduced CBG binding capacity and concurrently increased the levels of free corticosterone. So that's, remember, the primary rodent glucocorticoid. And we also found that co-treatment of neutrophil elastase with its endogenous inhibitor, alpha-1 antitrypsin, blocked this effect. And we found the same results in serum isolated from healthy human volunteers as well. But what do we know about how obesity influences neutrophil elastase? So I've just shown you some data from over a decade ago that showed that neutrophils are known to infiltrate adipose tissue under high-fat diet conditions. But what about systemically? And of course, to date, most of this research had only been conducted in male mice as well. So is there similar responses in female mice? Well, from our analysis, we found that circulating neutrophil elastase was not altered in our model of obesity, and that was in fact only elevated in adipose tissue and not, for example, in liver. And notably as well, we found that this effect was only observed in male mice, so we don't see any increase in neutrophil elastase in the adipose tissue of female mice. So again, suggesting sort of greater pro-inflammatory response in the adipose from these male mice. So the next question was to test the association between neutrophil elastase and glucocorticoid exposure. And so to do this, we utilized a mouse model lacking neutrophil elastase. So we fed male and female mice lacking neutrophil elastase and their wild-type litter mates a high-fat diet and did some initial metabolic characterization. Now, similar to previous reports, we found that in male mice, neutrophil elastase deficiency resulted in reduced weight gain, improved glucose and insulin tolerance, and improved insulin sensitivity as measured by HOMA-IR. But in female mice, we find this protective effect disappeared in terms of body weight and the other metabolic readouts. Of course, looking back, it probably shouldn't have been too surprising, as neutrophil elastase didn't appear to be elevated either in the circulation or at a tissue level in female mice. So knocking it out, if you like, was unlikely to have significant effects in this type of model. But of course, what about characterizing glucocorticoid action? So this is an area that had never been explored previously. So for this, we chose to proceed just in male mice, as they demonstrated clear genotype differences. And when we first looked in the circulation, we were a bit disappointed because we found removing neutrophil elastase didn't impact at all upon CBG binding capacity or indeed any measures of glucocorticoids, whether those be total or free levels. But a different story emerged at a tissue level. So in adipose tissue, we found that mice lacking neutrophil elastase had smaller adipocytes. But really interestingly, when we quantified corticosterone levels, they were also reduced. And this was in parallel to reduced expression of glucocorticoid responsive genes in period 1 and FKBP5 and increased expression of adiponectin. And notably in the liver, while lipid content in mice lacking neutrophil elastase was decreased, This did not occur alongside any changes in liver corticosteroid levels or indeed changes in glucocorticoid responsive transcripts in the liver. And really what we interpreted this as providing evidence supporting the idea that neutrophil elastase mediated cleavage of CBG increases local glucocorticoid exposure, and therefore in its absence, glucocorticoid signaling in adipose tissue is attenuated. Of course, so far these were only associations, and what we really needed to test was causality. And specifically, we want to ask the question as to whether the improved metabolic phenotype in neutrophil elastase deficient mice is actually driven by reduced glucocorticoid exposure. And so to test this, we removed the adrenal gland, so essentially taking glucocorticoids out of the equation, by performing bilateral adrenalectomies in both wild-type and neutrophil elastase knockout mice, and we had sham-operated controls for each genotype. And then we subjected them to the same high-fat diet and battery of metabolic tests, and what we found was that in the sham-operated animals, neutrophil elastase deficient mice once again showed improved glucose tolerance, but in adrenalectomized animals, this protective effect was lost. And the same pattern held for body weight gain and whole body insulin sensitivity. And so this provides strong evidence for an adrenal-dependent mechanism for how neutrophil elastase contributes to metabolic dysfunction. So obviously we were very excited by this, but as always, the next question, if you like, was, okay, well, does this regulatory axis exist in humans? And to explore that, we had to take a slightly different approach because neutrophil elastase deficiency is an exceptionally rare genetic disorder in humans. And to test this, we turned to a relatively common model, and that's alpha-1 antitrypsin deficiency. So if you recall, alpha-1 antitrypsin is the endogenous inhibitor of neutrophil elastase. So here, specifically, we studied heterozygous carriers of the most common SERPINA1 mutations. That's these S and Z alleles. And that's important because, crucially, these individuals are asymptomatic, so they don't have the liver dysfunction that we see in homozygous alpha-1 antitrypsin deficiency, which would lead to severe confounders. So studying these heterozygous carriers makes them an ideal model in which to study this neutrophil elastase CBG axis. So what we did was we used Generation Scotland to recruit 16 healthy heterozygous carriers alongside 16 age, sex, and BMI matched controls. And as shown here, the baseline characteristics were comparable between groups, with notable exception, of course, of a 30% reduction in alpha-1 antitrypsin levels in the carrier group. And of course, this correlated with increased circulating neutrophil elastase activity. Now, across 3 visits, we conducted 2 mechanistic studies. So the first was a tracer-based arteriovenous or AV sampling study to assess cortisol flux across tissues. The second was an assessment of the HPA axis. We did that using a receptor antagonist protocol, which we affectionately refer to as the CRASH protocol, or CRASH study. In the interest of time, I'm going to try and briefly summarize the results from both of these studies over the next few slides. Starting with our initial study, so to assess the dynamics of tissue glucocorticoid exposure, as I mentioned, we performed this arteriovenous or AV sampling across tissues, including abdominal subcutaneous adipose and skeletal muscle. We did this at regular intervals during steady state infusion of deuterated cortisol. So that's cortisol with 4 deuteriums attached, or D4 cortisol. And we used D4 cortisol because this enables quantification of both endogenous cortisol production, so that serves as an index of net cortisol production from all sources, while D3 cortisol production serves as a specific measure of cortisol regeneration by the enzyme 11-beta-HSD1. And together, this approach really allows quantification of in vivo whole body glucocorticoid appearance and clearance in parallel with glucocorticoid release across tissues. So it's a methodology that I could probably spend an entire presentation talking about separately, but today I'm just going to focus on some of the key results from these studies. Now, as expected, we found that D4 cortisol was in steady state by about 180 minutes. and in parallel, arterialized total cortisol, so this is endogenous cortisol, declined during the time course of D4 cortisol infusion. And there wasn't really a difference between our two study groups. But whenever we looked at parameters during the steady state, so from 180 minutes onwards, that's when we found some really interesting results. So we found that circulating free cortisol levels were higher in the alpha-1 antitrypsin heterozygous subjects, but no effect on cortisol. And interestingly, it wasn't associated with a decrease in CBG binding capacity either. But when we went on and performed subsequent regression analyses, we found that circulating alpha-1 antitrypsin and CBG binding capacity significantly predicted free cortisol. So really, in contrast to our mouse model, in humans we find that manipulation of neutrophil elastase via alpha-1 antitrypsin does in fact impact systemic free glucocorticoid dynamics. But of course, what about across tissues? So we set out to determine the net balance of cortisol and D4 cortisol across adipose and skeletal muscle. Unfortunately, due to technical difficulties arising from blocked cannulas in adipose tissue, that meant an inability to measure glucocorticoid dynamics across that tissue. Just a really tricky procedure to perform. But we were able to calculate net balance across skeletal muscle. And what we found is that there was a greater release of free cortisol across skeletal muscle in these alpha-1 antitrypsin deficient subjects. And what that does is really support this idea that there's an inhibitory role for alpha-1 antitrypsin in preventing local CBG cleavage across tissue and therefore glucocorticoid release. So after observing that the free circulating levels of cortisol were elevated in individuals with alpha-1 antitrypsin deficiency, or the heterozygotes if you like, whenever we infused a cortisol tracer, so an exogenous cortisol tracer, if you like, the next question was, okay, well, does this also happen under more physiological conditions, so without any tracer? And if it does, could this be linked to changes in central HPA axis regulation? So to test this, we use something called the crash test. So I mentioned this earlier. It stands for combined receptor antagonist stimulation of the HPA axis. Quite a mouthful, so crash is much easier to say. And essentially what this does is block both the main receptors that respond to glucocorticoids. So that's the glucocorticoid receptor and the mineralocorticoid receptor. And in doing this, we can assess whether the body's negative feedback loop, which would normally keep cortisol in check, is functioning properly. So we did this in a double-blind, randomized crossover design using a combination of RU486, which blocks the glucocorticoid receptor, and spironolactone, which blocks the mineralocorticoid receptor. And we had a placebo-controlled arm as well. And importantly, we tested this in the same participants that took place in the earlier study. So this is a timeline of the study itself. And initially, I just want to consider the glucocorticoid physiology at baseline, if you like, so in the placebo arm. And that's important because what we did here was confirm our earlier findings. So free cortisol was again significantly elevated in the alpha-1 antitrypsin deficient individuals. So that's shown in the solid grey and orange lines here, with no difference in total cortisol or CBG levels as well. And importantly, in this study, we were also able to obtain adipose biopsies. And this is where the story becomes even more clear, if you like, because adipose cortisol levels were significantly higher in alpha-1 antitrypsin deficient subjects. And this was accompanied by increased expression of glucocorticoid responsive genes like period 1 and SGK1, consistent again with increased tissue glucocorticoid exposure. Now, in terms of the HPA axis measures, so this is the crash element, if you like, as expected, combined GR and MR blockade increased total cortisol levels in groups, with no striking differences in HPA activation between alpha-1 antitrypsin heterozygotes and the controls, either in terms of free cortisol or CBG levels. And really what we think this means is that whatever impact lower alpha-1 antitrypsin levels are having on tissue glucocorticoid exposure, it isn't extending to central feedback sites, such as the the hypothalamus or the pituitary. So really no impact of alpha-1 antitrypsin deficiency on HPA axis activation. But what we have shown across these two kind of complementary human studies is that we've demonstrated that this subclinical, if you like, alpha-1 antitrypsin deficiency enhances both systemic and local cortisol activity. And the conclusion really there is that these results support the idea that this regulatory mechanism or axis, if you like, that was first identified in vitro is indeed at play in vivo in both mice, and it's conserved in humans as well, and it may therefore impact metabolic dysregulation. So in summarizing where we are at the minute, really this new strand of work that we've undertaken has served to build upon our current understanding of mechanisms that control glucocorticoid action. And it's identified yet another level of regulation, this time outside of the cell, that governs delivery of glucocorticoids to target tissues involved in contributing to cardiometabolic disease. Now, of course, the primary focus on this has been glucocorticoid delivery to obese adipose tissue, but this marriage, if you like, of neutrophil-mediated inflammation and glucocorticoid action is not specific to your classic white adipose tissue. And so in building upon this, we've really started to try and cast our net a bit wider, if you like, and ask if similar dynamics could be operating in other disease contexts and in other tissues. And so in my final few slides or so, I'd like to briefly share some snapshots of ongoing work around our investigation into atherosclerotic plaque development, so a known hotspot, if you like, for steroid hormonal interference. And this is important because a role for neutrophil elastase in atherosclerosis has recently been identified, with the group showing that mice lacking neutrophil elastase are protected from lesion development. And so we've been investigating how neutrophil elastase might control glucocorticoid action in this setting. Now for this we've been using a slightly different model of atherosclerosis, essentially a viral-induced model in combination with high cholesterol, which establishes atherosclerosis in 18 weeks. And to keep it relatively brief, in the interest of time, what we found is that there are indeed alterations in the neutrophil elastase CBG axis in this model. So we see circulating neutrophil elastase levels are increased, and this is in parallel to reduced CBG binding capacity. So in contrast to our high-fat diet obesity model, where there were no systemic changes in either of these parameters. Now, what we've done is investigate if removing neutrophil elastase is also protective in our model, as it was in the published ApoE model. And actually, much to our surprise, we found that neutrophil elastase deficiency had absolutely no protective effect. So clearly some important differences in the models here, really, that were to fully grasp, I suppose. But what it appears is that neutrophil elastase is dispensable for lesion development in our viral-induced model. And notably, we did also assess lesion development in mice lacking CBG. And this has given us some surprising results in that it appears removing CBG is actually protective in terms of lesion development. And we currently have a PhD student exploring the mechanisms underlying this. And it's particularly tricky trying to unpick these because, as you'll all be aware, one of the key challenges here is trying to quantify glucocorticoid levels or action at tissue sites. And in contrast to our mouse model of obesity, in which it was relatively straightforward to take a piece of adipose tissue, for example, and quantify glucocorticoids, extracting a lesion from a mouse artery to do this is really just not a viable approach. And so we've been working towards alternative approaches. And one of those is using mass spectrometry imaging. And this is very preliminary data, but quite exciting, we think. So if we consider a typical mouse lesion stained here, for example, with H&E, we can see the structure quite well. But with mass spectrometry imaging, defining the specific lesion area and not the vessel wall or the surrounding tissue is extremely challenging. And so as an alternative, we identified lesion-specific ions. And what we can do with those is use them as a sort of template to trace around the lesion area and then use that to quantify glucocorticoids, in this case corticosteroid, within just the lesion. So it's very limited data so far, very small n numbers, but we think it's really a really exciting technological advance that we can obviously use preclinically, but also potentially clinically as well, to assess glucocorticoid exposure in human plaques. So I'm aware I'm probably running close to my time limit here, so I'll just finish up by saying that these are really a number of settings, that there are a number of settings, I suppose, at the interface of inflammation and glucocorticoid-based endocrinology that are potentially regulated by this axis. But what really remains clear is that as the glucocorticoid and indeed the steroid biology field progresses, assessing the impact of changes in steroid binding globulins in parallel with not only total but free steroid measurements are likely to yield the next breakthroughs in our understanding of how our mechanisms underlie cardiometabolic disease. So I'm just going to end there by thanking the very many people here in Edinburgh, many of whom I've tried to highlight along the way, but also our close collaborators across Europe, in particular in Canada as well. You can find more information about our lab's research through the QR code there. But finally, I really just want to thank those who fund our research, notably the British Heart Foundation, who continue to play a huge role in driving this research forward. So thank you very much for your time.